All right. Okay, got it. Recording in progress. Well, it is my pleasure to introduce our speaker today. Our speaker is Dr. Cortes. She comes to us from Alabama, Birmingham. She's at UAB. It looks like she is currently an assistant professor there, has been there since 2019 in the Department of Cell, Developmental, and Integrative Biology. And she's only been there for a short time, but she has been, wow, productive. Lots of research, grants and awards and honors. And as you can see by the title of her talk, which is not in front of me right now, but she is studying the brain and how exercise affects the brain. She's done lots of work in the space of Alzheimer's disease. So it should be a really fun talk today. I'm excited about this. So just a little bit of background, Dr. Cortes did her PhD at the University of Chicago, where she worked on autophagy induction as a means of eliminating misfolded prion proteins before they become amyloid plaques in the brain. So Alzheimer's disease. She then moved out west to San Diego, University of San Diego, where she did a postdoc on the contribution of skeletal muscle autophagy to neurodegeneration in the context of triplet CAG repeat disorders for you people that are actually in the space of Alzheimer's and know what that means. Dr. Cortes has her own lab now since 2019 at UAB. Her lab is studying brain aging, especially in the context of organismal physiology and neuropathology. And she looks at exercise as an intervention against age-related neurodegenerative diseases. Wow, that was a mouthful. So happy to introduce you today. And without further ado, I will turn the Zoom stage over to Dr. Cortes.
Thank you so much for that amazing introduction. And as I was saying earlier, I'm really bummed that I couldn't be there in person, but hopefully next time. And so I'm going to go ahead and share my screen with you guys. Give me 1 second here. Are you seeing the correct view? I always have to figure out if we're doing presenter or not.
Yeah, yes, looks great.
Awesome. Thank you so much. So today I'm going to be telling you a little bit about what we've been doing since we started my lab in 2019. And actually, if you're keeping track, we actually had 3 months before COVID hit. So it's been a fun experience to run a lab in the middle of a raging pandemic. So if anybody wants to chat about that after, I have, opinions on it. But today we're going to be talking about, my 3 favorite things, as the song goes. And I titled my talk, Geroprotectionon on the treadmill, exercise, CNS aging, and Alzheimer's disease. But in reality, as I'm sure many of you in the audience know, exercise is good for you and good for pretty much anything. And so we truly believe that some of the approaches we're taking are broadly applicable to multiple diseases. So we're going to, and by the way, please feel free to interrupt me with questions. If you raise your hand, you'll pop up on my screen, and I'm happy to open the floor for you to ask. So in my lab, we're interested in understanding the mechanisms that underlie the neurobiology of aging. So how do we go to a young, healthy brain like this one on the left to an aging brain like this one on the right? And in particular, we focus on 3 distinct hallmarks that occur in the aging brain. These are actually hallmarks that occur in most aging tissues, but they're particularly prevalent in the aging central nervous system. And these are proteostasis and neurogenesis, or stem cell function, and inflammation. So during aging, what we see is a decrease in proteostasis, or protein quality control pathways, that is believed to underlie the accumulation of these age-associated protein inclusions, including those found in Alzheimer's disease, Parkinson's disease, and other age-associated neurodegenerative disorders. We also see a decline in neurogenesis activity. So when I was in grad school, I was told that the number of neurons you're born with are the same ones you're going to die with, and that is actually incorrect. There is active neurogenesis that occurs in the hippocampus, and this process is significantly impacted during aging. And there's also an increase in inflammation. And so if you are in the aging space at all, you may have heard about the hypothesis of inflammaging. The older you are, the more inflamed you are. And if you can delay that inflammatory processes, you can delay some of these hallmarks of aging. And so in the top of this diagram here, we have what we call a young brain. And at the bottom here, we have an old brain with protein inclusions accumulated, neural stem cells that are dying off, as well as increased inflammation, leading to the age-associated structural changes that we see in the brain, which include cortical and atrophy, as well as brain atrophy of other brain regions, including the hippocampus, as well as enlargement of the ventricles shown here. So in the field of brain aging, we've also come to understand that a lot of these processes contribute to the increased risk for the development of neurodegenerative diseases that we see in older populations. And although today we're going to talk about Alzheimer's disease, like I mentioned in the beginning, a lot of these same processes are altered or accelerated during Parkinson's disease, vascular dementia, so on and so forth. So in the field of aging, we know that one of the most powerful behavioral interventions against all of these hallmarks is actually exercise. And although the mechanisms responsible for these benefits are still very much being studied and we are still discovering them, they all tend to go through this increase in the levels of hippocampal brain-derived neurotrophic factor. So this neurotrophic factor allows for neuronal survival and is associated with decreased inflammation, increased neurogenesis, and improved protein quality control in the aging brain. So recently, this is a review that we published out of my lab during the year of reviews, that was 2020. What the field of exercise has come to realize is that actually during exercise, there's an accumulation of what we call circulating factors in your blood that can come from multiple tissues, but currently the ones that are most characterized appear to come from muscle or from liver. And the enrichment of these factors in circulation is believed to drive multiple benefits in the brain associated with behavioral exercise. And so again, the mechanisms that these factors act through are still unknown. We believe some of them may potentially act at the blood-brain barrier. Some of them may actually cross the blood-brain barrier. But ultimately, all of them lead to this increase in BDNF production in the hippocampus, as well as the rejuvenation of the central nervous system, which includes the phenotypes that I mentioned earlier. And through fascinating plasma transfer studies, we've now demonstrated that you can transfer the benefits of exercise by simply taking the blood from a runner and putting it into a sedentary mouse. And so this is where this idea that there's something in your blood that can provide the benefits of exercise provides a unique therapeutic targeting opportunity, right? If we can identify these factors, maybe we can develop drugs that mimic their effects, and we can provide the benefits to exercise to both aging populations that necessarily sometimes cannot exercise, and essentially allow all of us to get the benefits of exercise without necessarily having to get on a treadmill. So as demonstrated in this figure, one of the most powerful responders to behavioral exercise when you get in the treadmill, when you go work out, when you go running, when you go dancing, is actually skeletal muscle. And, this is simplifying decades of work, and there's still a lot between these arrows that we don't know, but we do know that exercise can counteract a lot of the aging phenotypes locally in skeletal muscle. It can activate mitophagy, and the quality control process, both proteasome and autophagy activity are activated, leading to preservation of proteostasis and then improvement of metabolic health. I think I don't need to tell this audience how important skeletal muscle is for the control of glucose signaling in the body and glucose metabolism. And so what happens at the biochemical and cellular level in skeletal muscle during endurance exercise? That's what I'm going to be focusing on today. So this is associated with cardio, so running, dancing, getting on the treadmill, different things happening muscle when you actually do weightlifting, which is resistance exercise. So there's some measure of muscle hypertrophy that happens during endurance exercise. There's fiber type switching. As this audience knows, muscle is composed of multiple kinds of fiber types. And depending on their metabolic and bioenergetic requirements, they can switch from type 1 to type 2. And within type 2, there's 2A, 2B, and 2X. And during exercise, these fiber types can switch. As I mentioned, there's activation of proteostasis in skeletal muscle. There is increased biogenesis of mitochondria. You can imagine if you're asking muscle to continually contract for extended periods of time, that consumes a lot of the ATP reserves that we have. There's also increased capillary density and blood flow that occurs. If you open up a mouse that's been running for a couple of months versus a mouse that's sedentary, you can actually tell who's who. The muscle is redder because they require increased blood flow to deliver oxygen for ATP production. Increased oxidative capacity of the mitochondria that exist in the muscle. And as I mentioned, there's a reduction in the hallmarks of aging. And so, and there's also changes in the secretome. And again, I'm sure this audience may be familiar with it already, but skeletal muscle is actually an endocrine organ. It secretes multiple factors into circulation, proteins, metabolites, microRNAs. The muscle is in constant communication with the rest of the body. And we believe some of these factors are the ones that can target the brain and provide some of those benefits into the aging central nervous system. And so the question I was interested in when I started my lab is, if proteostasis is this important during exercise, what happens if we activate proteostasis without behavioral exercise? And in particular, how does that affect some of the central nervous system effects that we see under these conditions? And so to answer this question, I took advantage of one of my favorite proteins. So as was mentioned in the introduction, in grad school and as a postdoc, I was, in the field of autophagy, and I fell in love with it, and I'm still in love with it, even though it's very painful to work in it sometimes. And so this is my favorite protein, transcription factor EB, or TFEB for short. And this is a master regulator of lysosomal function in autophagy. As you can see in this diagram here, in its inactive state, TFEB resides in the cytosol. Upon activation, it moves into the nucleus to drive the transcription of a variety of autophagy and lysosomal gene targets. And if you're familiar with the autophagy pathway at all, you'll notice that it's able to modify every single step of the pathway, all the way from initiation to cargo recognition, to fusion of the newly formed autophagosomes with the lysosome, as well as degradation of the cargo. And this makes TFEB one of the most powerful tools in the arsenal of autophagy activators, both in vivo and in vitro. And so I generated an inducible human TFEB transgenic mouse. And so I placed the human TFEB transgene downstream of the stop codon, such that there is no exogenous expression of the transgene unless I cross these mice with a Cre recombinase that can remove this entire operon right here and allow for the promoter to actually drive the transcription of my transgene. So this is what I like to call a Cre-on system. And so to drive the expression of this transgene to my tissue of interest, which was skeletal muscle, I crossed these flock-stop TFEB mice with a human skeletal actin Cre driver line. And this Cre only expresses in skeletal muscle. So the combination of my transgene requiring Cre activity for expression and the Cre activity only being present in skeletal muscle results in this double transgenic mouse line where there is activation of my transgene only in tissues of myogenic lineage. So to confirm that, we first performed immunoblotting of skeletal muscle lysates, and I hope you can appreciate here in B. On the right, we have TFEB HSA Cre transgenic skeletal muscle lysates displaying prominent expression of my exogenous transgene for TFEB. And depending on the age and the sex that you look at this, we have anywhere from 3 to 5-fold expression of the endogenous transgene of the exogenous transgene over the endogenous transgene. And this is specific to muscle. We don't see this in the brain, importantly, for some of the data I'm going to show you later. These are the same individuals actually shown here, or in other highly active metabolic tissues such as liver. We've run both kinds of fat, we've run kidney, we've run heart. We only see this in skeletal muscle. So we had confirmed muscle-specific expression of my TFEB transcription factor. But the next question is this actually active? And so we performed qRT-PCR analysis of skeletal muscle RNA lysates now, looking for the expression of classical TFEB lysosomal and autophagy gene targets. And I hope you can appreciate here in the black bars, significant overexpression of these targets compared to the non-transgenic litter-mate controls for the TFEB HSA-3 transgenic muscle tissue. We also confirmed that this translated to elevated protein levels of some of these targets. So we looked at lysosomal proteins such as LAMP1, which is a structural lysosomal protein and a known TFEB target. You can see it over here on the qRT-PCR, as well as cathepsin D, which is one of the enzymes that resides in the lysosomal lumen and processes the cargo that's delivered to the lysosomes. And it's also a TFEB target. It's shown here on the qRT-PCR. And both of these are also significantly elevated in the double transgenic skeletal muscle, suggesting that yes, we had not only overexpressed our transgene, but we had actually achieved functional activation of the TFEB transcriptional network. We also confirmed some mild but consistent elevations in autophagy. So to examine autophagy, one of the gold standards is to examine the processing of this protein called LC3 into its active isoform LC32, and then increased ratios of LC32 over LC31 are usually suggestive of an accumulation of autophagosomes and autophagy activation. And we were able to detect that in the double transgenic skeletal muscle, which suggested that through overexpression of TFEB, we had maintained or activated autophagy to a degree that was maintained throughout the lifespan of this animal. And so to kind of get at that, we began to look at our mice as they got older and older. And at 16 months of age, which is about midway through the lifespan of a mouse, depending on your mouse room conditions and the mouse line you're looking at, mice usually live up to 3 years usually, so that would be 36 months. So you can see this is kind of halfway there. But at this point in time, many tissues, including skeletal muscle, begin to show the classical hallmarks of aging. And one of the hallmarks of aging is the accumulation of protein inclusions, as I mentioned in the beginning. And so what you can see here is a cross-section of quadriceps muscle where the fibers are coming at you from the screen. So you can see, for example, one muscle fiber is right here, outlined with DAPI in blue. And when you stain for markers of protein aggregates, such as P62 and ubiquitin, at this age, you see the accumulation of these big, inclusion-looking structures is quite abundant in the P62 stain. And when we compared an age-matched littermate that was a TFEB HSA overexpressing animal, I hope you can appreciate the difference. That again, this is a cross section, the fibers are coming at you. There's a fiber right here, for example. The accumulation of these structures is significantly reduced. And this, consistent with this blot for LC3, suggested that we had preserved protein quality control in the skeletal muscle of these animals and prevented the age-associated accumulation of these structures in muscle. So what else were we finding? Well, let's look at the architecture of the muscle of these animals. And so we did H&E staining. And again, you begin to see classical markers of muscle aging. So one of them, once again, these are cross sections of quadricep muscle. one of the classical hallmarks of aging in muscle is what we call the decompactation of the muscle fibers. So they should be tightly packed together with no empty spaces between them. Whereas as the aging begins to occur and some of the muscle fibers begin to deteriorate and waste away, you start to see openings as this begins to happen. You also see the mobilization of the myonuclei from the periphery where they should reside into the inside of the fiber, and that's outlined here in the yellow dots. And then you also see the accumulation of these structures that stain positive for trichrome, which we believe match with the P62 and ubiquitin staining that I showed you before. So these are multilamellar, multi-protein enriched inclusions that are accumulating as autophagy and proteasomal activity begin to decline in this aging skeletal muscle. And again, I hope I don't have to tell you that they're different in the muscle TFEB, in the muscle overexpressing TFEB. These are again littermate controls, age match and sex match littermate controls. The appearance of this muscle TFEB expressing muscle, it's much more consistent with a 6-month-old muscle as opposed to a 16-month-old muscle. So what else is going on in locally at the skeletal muscle level? We're going to spend a lot of time on the muscle in this talk because I know this is probably something you guys are really interested in. So we looked at some mitochondrial proteins by Western blot shown here on the left. And as you can see, we're able to detect significant upregulations of multiple components of the mitochondrial network in TFEB overexpressing muscle shown here. And then when we performed electron microscopy imaging, and again, I hope I don't have to tell you, but we also saw significantly enlarged mitochondria in the gastrocnemius muscle and the TA and the quadricep, I believe we did, in the muscle overexpressing TFEB compared to the age-match controls. So what this suggested was that there was an increased mitochondrial load happening in the muscle of these mice. And so we wanted to examine mitochondrial function. So we performed Ouroboros studies at the Barshop Institute Nathan Schock Center. And we submitted multiple samples, but as you can see here, this is the soleus respiratory activity. And for those of you that have never done Ouroboros analysis before, it's very similar to the seahorse, if you've done that one. Essentially, you measure maximal mitochondrial activity in the presence of multiple inhibitors. And that's what each of these bars represents down here, inhibition of complex 1 and 2, so on and so forth. And what we saw in control animals is the expected age-associated decline in mitochondrial activity. So we're comparing here a middle-aged mouse that's about a year old versus an age mouse that's 2 years old. You see there's significant reductions in the maximal activity of the mitochondria in the soleus. And when you compare that to a muscle TFEB expressing animal, again, the same ages, what you can see is that this activity does not go down. So it's at baseline, it's not higher than the controls, but rather what we're doing is we're preserving mitochondrial activity, probably through preservation of mitophagy, and preventing this age-associated decline in the mitochondrial activity in the muscle of these mice. What else does muscle do? As we mentioned earlier, muscle is heavily involved in glucose metabolism. So shown here on the left is some glycogen staining. And the deeper the purple, the more glycogen the fibers have. And the muscle TFEB tends to be purplish compared to the age-match controls, suggesting that they may be accumulating more glycogen in their fibers. We also were able to detect increased levels of GLUT4. This is total levels. We haven't done the membrane-bound levels of GLUT4 yet, but at baseline, there is increased expression of GLUT4, the central glucose transporter that mobilizes glucose from circulation into the muscle fiber. And again, when we look at the levels of glycolysis enzymes, multiple members of this pathway are also significantly increased in TFEB expressing skeletal muscle, suggesting again, not only do they have more mitochondria and better working mitochondria, they also have more fuel for the mitochondria at some point. And so because of this, we were really interested in testing exercise tolerance. So can these mice run longer? And the answer is actually yes. So at 6 months of age, which is a fully developed animal and in the field of aging is young actually, these animals, both the females, both the male and female TFIB expressing animals can run for longer and can perform more total work than their age-matched littermate controls, consistent with this idea that they may have more available fuel. But what was really interesting is that this difference was preserved, at least in the females, for this older age of 18 months of age. 18 months, again, is like midway through, they're entering what we call an old age. And as you can see here, the TFIB females can also run for longer and perform more total work than their age-matched littermate controls. We didn't see that difference in the males, but I'm not sure if this is because we didn't have enough males at the time to get the difference or because there is a sex-specific effect here. We're currently heavily investigating those two options. So what else is going on with these mice? Well, we started noticing that the older they got, at about 6 or 7 months of age, they started to significantly separate from their littermate controls as far as their weight. And so we were really trying to figure out what is going on. Why are they heavier? And so our first hypothesis was, well, it's because their muscle is bigger. But actually, we were not able to detect any significant differences in multiple muscle types that we looked at when we compared the TFIB animals to their age-matched controls. But rather, this difference in total weight appears to be driven by an increase in the white adipose tissue. So these mice are not obese by any means, they just have more white fat. They also have smaller livers. There appear to be no differences in their brown adipose tissue, and they have a slightly enlarged heart. And so interestingly, all of these things are things that are similar to what you would see if you had running mice. So importantly, all of these animals are just sedentary, right? The only difference they have compared to the controls is the expression of the TFIB transgene. So I see a question from Dr. Dillon in the chat. She asks, would you expect to see additional benefits with a combined aerobic and resistance exercise protocol? Yeah, so that's a question that I get all the time. We are actually running a lot of these animals right now at our exercise core, so I'll be able to answer that question in a couple of months. But I'm really interested to see if we'll see even a further, further benefits in the context of the TFIB background, or if actually they'll, what I like to say, they'll collapse into toxicity. They'll have too much autophagy. So I don't know. So, but we're excited to see the results from those studies for sure.
Thank you.
So, but this work told us that there are systemic things happening in these mice that are independent of the local changes in muscle that we're seeing. And so we were wondering if this was driven through changes in the skeletal muscle secretome, right? Again, muscle secretes cytokines and microRNAs and metabolites, and they're all collectively called myokines. And so just to see what was going on, we performed a directed analysis of classical myokines, as I like to call them. There's many, many, many more than these. These are just some that respond to exercise, number one, and number two, have been shown to target the central nervous system because, spoilers, I don't know if you guys have realized this, I'm actually a neuroscientist. So I'm really interested in how the muscle and the brain talk to each other. But essentially what we saw is under baseline conditions, the muscle of the TFIB muscle had reduced levels of myostatin, which was good. As you guys know, myostatin is a marker of skeletal muscle stress. So the fact that it was reduced in our animals is good. We have increased expression of interleukin 15, increased expression of FNDC5, and some indications of other things that are going on. And now these were young animals. We've expanded this analysis to the older ages as well. And there's some really interesting thing happening that are age associated, number one, two, sex associated, and three, genotype associated. And so that's currently what my lab is actively working on, trying to figure out if these changes in myokine signaling are responsible for some of these systemic effects that we see. I can see a couple more questions in the chat. Dr. Potter is asking if I see differences in white adipose tissue. Yeah, so as I showed earlier, we see that they have larger white adipose tissue deposits. I don't know what it means. I just know it's there. And yes, absolutely, the muscle talks to the adipose tissue all the time. And so it's really possible that there's a phenotype happening there that we're just currently not able to tackle quite yet.
Yeah, I was actually just curious if you looked at the depot, like if it was sub-Q versus visceral.
Oh, which one? So that one that I showed was inguinal fat, I believe. Yeah. And so we're expanding that for, the suprascapular and the subdermal and all of that good stuff. We just got some pilot money to do that. So yeah, absolutely. Amadeo asks, is there a synergistic effect of nutrition and exercise on TFib activity? And is TFib stimulated through mTOR-dependent and independent pathways? Yes. So if you notice in one of the cartoons that I showed of TFib function, it's phosphorylated, and mTOR is the central kinase, as is with everything, right? So mTOR directly phosphorylates TFib. There are other kinases that do it too. I believe ERK1 and 2. There's a couple of MAP kinases that do it as well. So there's also mTOR independent pathways. And exercise alone activates TFib in muscle. And I don't know if anybody has looked at nutrition states, like changes in diet, but I bet you a response as well. It's such a central node for connecting autophagy to metabolism that I would be surprised if it didn't respond to nutrition derived changes. Cool. So let's keep going. So just checking my time here. Okay, we're good. So then, oh, did another question pop in? Oh, yes. So Brian asks, during exercise, are there differences in expression between myokines such as cathepsin and urisin? I see where you're going with this. Yes, there is. So cathepsin B actually is one of the myokines that Dr. Van Praag, who's one of the pioneers in the role of exercise-activated neurogenesis, was able to discover is one of the myokines that comes out of muscle during running in particular, and appears to be able to cross the blood-brain barrier and activate neurotrophic signaling in the brain. Urisin is another one. If you're in the field of metabolism, diabetes, it's one of the ones that communicates between muscle and fat, but now it's been found to apparently also communicate muscle or peripheral signaling with the brain. And so exercise activates both. They're also activated in my mice as well. And then you're asking if neuroprotective benefits favor regulating neuroinflammation or reducing levels of amyloid beta peptide. Yes. So give me a couple of minutes. We'll get to that. Absolutely. So then to kind of summarize this muscle and kind of neck down information that I've given you, the skeletal muscle adaptations that we see in the muscle TFib expressing mice are actually similar to that seen during endurance exercise. So we don't see any muscle hypertrophy, at least, by wet weight. And we've done the lean and fat mass analysis as well, and we don't see any differences there. We're doing the fiber type switch analysis right now, and there's some indications that there's something going on, but I need more data before I confirm that. But we do see increased proteostasis. We have increased mitochondrial biogenesis and mitochondrial function, especially throughout aging. I didn't have time to talk about it, but our mice are redder too. You can tell them what the genotype is when you open them up because they have increased capillary density in the muscle. There's increased oxidative capacity. We showed that with the Ouroboros studies, and there's reduced hallmarks of aging locally in the muscle. Oh, I think I see another question. Oh yeah, it was just a response to the previous. Okay. So then, like I said, and there are similar changes in the myokine profile, and we're currently comparing essentially what comes out of muscle during exercise, what comes out of muscle in my TFib animals, and can we use this as a prioritization tool right? Because hundreds of things come out of muscle during exercise, but what is actually driving the benefits is what we're trying to get after. So like I mentioned earlier, I'm actually a neuroscientist. And so I was really pushing my lab to go look at the brains of these animals, and we did. And so we looked around 18 months of age. As I mentioned, that's entering what we call elder stages. If you were to translate this to human terms, it would be about, 50 to 60 years old in a human. And what we saw is that by qRT-PCR analysis of RNA brain lysates, we were able to detect significant upregulations of master regulators of proteostasis. And this included HIF1A, heat shock factor 1, and sirtuin-1, which was really interesting to us because, as you may know, sirtuin-1 and heat shock factor 1 are master regulators of aging-associated pathways. We also saw some elevations of individual members of other protein quality control pathways, including chaperones, both cytosolic and mitochondrial, and some indication of activation of autophagy and lysosomal genes in the brain of these aging muscle TFib expressing animals. So remember, my transgene is not expressed in the brain, and I showed you the evidence for that in the beginning. So this, we believe, is driven by a muscle effect signaling to the brain and activating proteostasis. And especially at these older ages, this suggests that we may be also maintaining brain proteostasis in addition to maintaining muscle proteostasis. And so to look at that, we focused, as I mentioned in the beginning, on the hippocampus, which is a region that atrophies with age and is one of the sites of multiple age-associated neurodegenerative diseases. And so we looked at lipofuscin. So lipofuscin, as the name suggests, is an autofluorescent polymer that accumulates in lysosomes. So as lysosomes age, their ability to actually degrade the cargo that is delivered to them begins to decline, and that leads to the accumulation of essentially trash. And so if you've ever looked at any kind of aging tissue, you've probably seen a lot of autofluorescence signal, and it's driven by this phenotype. And also, if any of you have elderly relatives, or some of us are starting to develop those now, and you have age spots in your skin, that is lipofuscin accumulation in your melanocytes. So that's how prevalent this phenotype is. And it also happens in the hippocampus, and especially in the dentine gyrus of the brain, which is this banana-shaped structure right here. You can see the banana is coming this way in this orientation, and lipofuscin is shown here in red. And again, I hope you can appreciate the difference. The dentine gyrus is now oriented going upwards this way, but the accumulation of this lipofuscin is markedly reduced. So then we said, well, what else is happening when other hallmarks of aging are maybe being altered in the brains of these animals? So we looked at levels of BDNF expression in the hippocampus, and we saw a very significant upregulation of BDNF in the hippocampus of these muscle TFib expressing animals. And then we said, well, if BDNF is up, is neurogenesis changing too? And so this is a preliminary analysis of neurogenesis signaling that we looked at, where you inject animals with EDU, which is a thymidine analog that gets incorporated into dividing cells. And so they'll get labeled with it. And then you can count the number of cells that are positive for EDU, again, in the hippocampal subventricular zone and the dentine gyrus. You can see the banana, the shape of it very well here. And so these are the cells that we count that are positive for EDU, and double cortin, which is a marker of this dividing cell has actually committed to divide and commit to a neuronal lineage. And we saw significantly elevated levels of these cells in the hippocampus of 2-year-old mice. So they're very old. They usually have very few, if any, of these cells. And we were able to detect increases when they were expressing TFib in their skeletal muscle. And so does this translate to functional improvements, right? Because that's the ultimate goal, is can we use some of this to actually improve cognitive function in obviously patients, but for now in my mice. And so we ran some behavioral assays at 18 months of age at the Scripps Neuroscience Behavioral Core. And the first one we did is the Barnes maze. For those that don't know it, essentially it's an exposed platform that has one escape tunnel. And what you're asking the mouse to remember is the location of this escape tunnel. Mice that don't remember where it is will wander around looking for it in between sessions, whereas those that do remember will start going to that section very quickly after a couple of days of training. And what we were able to see is that both male and female muscle TFib expressing animals committed less errors and escaped a lot faster from the muscle. Escape is when they go into the hole, than their age-matched non-TFib expressing littermate controls. So we ran these studies after, of course, having done multiple analysis on locomotion. Both these animals move the same. Visual acuity, these animals see the same, and other parameters that could perhaps affect this. So we truly believe this is a cognitive effect that we're achieving here. And to look at that, we performed a different kind of test, which is called the novel object recognition. And in this one, during the training setup, you present the individual with objects that it can familiarize itself with. And during the testing setup, you give it one of the old familiar objects and a novel object, hence the name of the test. And the goal here is that you're asking the mouse if it remembers that it knows what this is, but it doesn't know what this is. So it should have a preference to investigate that one. And indeed, that's what we see during the trial test when we introduce the novel object C into the test. The muscle TFib expressing animals have a higher preference for the novel object than their age-matched littermate controls. And that's also shown down here. So before we switch, I see that we have a couple of questions. Let's see. So Clayton asks that TFib can regulate many autophagy markers. Is there a specific autophagy target that TFib is regulating? Did I measure autophagy flux? Yeah, so TFib is a master regulator of hundreds and not thousands of genes. It's called a CLEAR network, and it's growing every day as new targets are identified. We are doing RNA-seq on these muscles right now, but in reality, it's just a tool for us to activate autophagy right now. So we haven't measured autophagy flux in vivo because it's quite tricky to do. We have done it in vitro. We've grown myoblasts from control and TFib positive animals. And there we do see, massive upregulation of autophagy flux, doing the classical, inhibition of lysosome and then doing the ratio plus minus lysosomal inhibitor and all that good stuff. So yeah. Then regarding GLUT4, the data is, oh, thank you. And so you're wondering if there's a relationship between autophagy early late endosome and the organization of the Golgi complex. And could I comment on that? Absolutely. So in the field of autophagy, we often think about it as just a recycling pathway, right? It's taking things from the lysosome and from the cytosol and delivering them to the lysosome for degradation. But actually, if you think about it, autophagy is simply a membrane remodeling pathway. It takes membrane, we know now, from the ER and the omegasome and all that cool stuff, and creates a new organelle called the autophagosome by essentially building membrane around the structure. And so you can imagine that it's heavily involved with vesicle trafficking back and forth. And so if you think about, the GLUT4 containing vesicles, residing right below the membrane, and then being triggered to fuse, it'll be fascinating to see if that's why we're seeing what we're seeing, right? Is it just because we have more vesicles, or that are they better primed to move and mobilize and dock with the membrane? Or is that they're just more GLUT4 packaged on them? All of that, I think, would be a fascinating line of investigation, but I don't have an answer for you on that right now. Then Dane is asking that HIF1A, which is one of the factors that we saw upregulated in the brain of the aging muscle TFib mice, is mediated by hypoxia. Absolutely. And do I speculate if TFib is incurring some sort of cellular hypoxia? You know what? I hadn't thought about it that way, but if you bring in this idea of mitochondrial hormesis, right, being important for resistance to some of these things, then I think that might be something we want to take a look at. Really good point. Yeah, thank you for that suggestion. Dusty is asking if the hippocampal effects that I showed are BDNF dependent. So I haven't done the blocking experiment yet. We do see upregulation of BDNF in the hippocampus of these animals, so we know it's getting activated. But is it required for the effect? We haven't tested it, but my expectation would be that yes, it is. There's a beautiful paper on what is it, Science, I think it is. It could be Nature, for where they ran animals and then they saw the expected activation in neurogenesis, and then they ran them again and they blocked their ability to induce BDNF, and then the neurogenesis activation went away. And so that requires, that suggests that BDNF is required for these benefits to manifest. And so I would hypothesize that yes, it is dependent on BDNF activation, but I haven't tested it yet. Okay, so that was healthy aging, as I like to call it. Let's get into disease now, right? So these are the hallmarks of aging. And if you've ever read an aging paper, you've probably come across this picture. We actually have competitions in the field of whether you can go through an aging talk without seeing this wheel. So congratulations, I failed. But essentially the hallmarks of aging listed here are 8 shared phenotypes that occur all across aging tissues, including the brain. And interestingly, when you look at Alzheimer's disease brains, at least 7 of them are also found in Alzheimer's disease brains. And so what this suggests is that pathways that can protect against healthy aging may also be able to protect against Alzheimer's disease. So we did the obvious experiment. I crossed my mice to Alzheimer's mice. So after a lot of work, a lot of crosses, and a lot of breeding, luckily we had COVID to wait out. And so we just set up breedings and waited. We crossed the muscle TFib expressing animals to 2 different models of Alzheimer's disease associated pathologies. One is a tau model where they carry the human tau protein carrying this mutation. And then the second one is a plaque-based model called the 5XFAD transgenic mouse. And the only things you need to know for this particular talk is that the tau mice develop tau pathology, but no plaques. And the plaque mice develop plaque pathology, but no tangle pathology or tau pathology. So we were trying to separate, these are the 2 hallmarks of Alzheimer's brains. Can this help with one or both or neither? And then also both of these models are characterized by neuroinflammation. So we're going to get to whoever asked that question earlier. Because we know one of the multiple of benefits of exercise, but one of its most powerful effects is it can decrease inflammation in the periphery. And a lot of studies suggest it can do the same in the brain. And so that's what we wanted to take a look at. So first, I'm going to show you some data on the tau model. So first here on the top right, I'm showing you that in the quadriceps of these tau mice, I have successfully achieved expression of my transgene in the context of the TFib HSA-3 background. Obviously, the tau mice in the wild type background don't have TFib in muscle or don't express my transgene. Again, this is the denti-gyrus of the hippocampus. The banana is pointing up in this picture and pointing down in this picture. And we stained for the 3 markers of pathology that accumulate in these mice. One is phospho-tau, which is in green, which you can see forms these, very, very big inclusions inside the neurons. And we also stained for markers of neuroinflammation. GFAP is a marker for astrocytes, and that's in white. And then IBA1 is a marker for microglia, and that's in red. And even if you've never seen one of these pictures, I hope you can appreciate that there's a lot of red, and you can even see the white because it's superimposed with the green. There's a lot of green too, right? And then when you compare that to an age-matched littermate that actually had muscle TFib expression, I hope you can appreciate the difference. There's a lot less green, a lot less red, and a lot less white, suggesting that we had potentially reduced the accumulation of tau, which is the pathological marker, and concomitantly also reduce the accumulation of activated astrocytes and microglia, which drive the chronic neuroinflammation present in this model. We were also able to rescue the declining levels of BDNF in this mouse. So this is the wild type littermates compared to the single transgenic tau animals compared to the triple transgenic tau in the TFIB background. And so we were able to elevate the levels of BDNF in the hippocampus. And also somebody asked about FNDC5 and Orison. We were also to almost rescue that. We're letting these animals age a little bit more now. I believe this was 6 months of age. So now we're collecting the 9 months when the pathology has really, frankly, destroyed the brain, and see what we can see there. So then what else is happening at the transcriptional level? Are we able to change the transcriptome of the brain that may be underlying some of these benefits that we see in the pathological stains? And so to do that, we ran the hippocampi of these animals through the nanostring encounter Alzheimer's disease panel, which is a compilation of 700 different genes that are known to change during Alzheimer's disease. And we ran 6-month-old animals. And as you can see here on the orange dots, these are individual RNA, hippocampal RNA lysates of single transgenic tau animals compared to the tau in the TFIB background. So we are seeing significant changes on transcription and splicing go terms, essentially, as well as some changes in lipid metabolism. And as we kind of had seen with the BDNF, some changes in trophic factors as well. So there's definitely transcriptional remodeling that's happening in the hippocampus of these animals, which is super exciting for us. And we're currently pursuing what this could possibly mean mechanistically. And then this is one of my final slides. We've now finally gotten the triple transgenic 5XFAD animals. These are the ones that are plaque-based. And so once again, I'm showing you the hippocampus here, the banana. In red, I'm showing you the plaques. And so you can see that we haven't eliminated the plaques, but we think we have actually reduced the amount that accumulate by 4 months of age in the brains of these animals. And we're also able to correct some of the pro-neuroinflammation signaling that's happening in the brains of these animals, including corrections in interleukin-6, interleukin-1 beta, and other members of the microglial response. And so to summarize everything that I've told you today, by overexpressing TFIB in the muscle of these mice through their lifespan, we've actually improved muscle proteostasis. We believe we've improved muscle metabolism, and we've preserved muscle health throughout aging. And we believe this leads to the preservation of a young muscle secretome, the way it communicates with the rest of the body, and in particular with the brain. And we believe this may be a new strategy to improve neuroprotection of the aging brain, as well as in the context of age-associated proteinopathies. And so with that said, I'd like to thank the people that made this work possible. This is my lab. This was the first time we were able to meet in person. We're all triple vaccinated at this point. We all got boosters too. And so super excited to work with this amazing team, as well as our funding. And I'd be happy to take questions.
Wow. Well, as we usually do, I'll have everyone unmute, if you will, and clap for our speaker. That was really an amazing talk. So thank you for that.
Can you stop sharing? Yeah, so we can.
Yes, there we go.
Yeah. And we can open it up for questions. I think you probably, I'm not seeing the chat, but I think you probably addressed all of those. You did a wonderful job kind of going back and forth, watching that chat, answering those questions, getting right back at it. So that was great. That was really, really interesting. So I am particularly really interested in those sex differences that you alluded to. And so that's one, I don't want to monopolize that. I have a lot of questions. That was really interesting. Can you talk a little bit about those sex differences?
Yeah, so essentially what we're seeing is that both sexes are equally protected, if you will. We see similar activation of everything that we sort of look at when they're young. When we run the same studies when they're old, they begin to separate. Okay. So we see a reduction in neuroinflammation only in the aged females, but not in the aged males. And I
Okay.
don't know why.
Okay.
And so that's why a lot of my data is parsed out, because I want to make sure that I can see a sex effect when it's happening. And you guys know that there's sex differences in responses to exercise, there's sex differences in Alzheimer's disease presentation, and there's sex differences in aging, not just of the brain, but you know, as a whole. And so, yeah, so I'm super interested to kind of dive deeper into that.
Have you looked at estrogen receptor expression anywhere in your brain cells or?
I actually haven't, and I think that would be kind of like an obvious one, because it does appear to be that the females are more protected than the males the older they are. And so that's a really good suggestion. Yeah.
Cool.
So I think Clayton has their hand raised.
Yes. Thank you, Dr. Cortes. Amazing talk today. I have two questions for you. Have you measured specific force in the skeletal muscle of your model? And also, it has been shown that if you activate BACLIN-1 in aged mice, you can clear, for example, protein aggregation like alpha-synuclein, and also improve the cognitive function of those mice. Have you measured also cognitive function in your model?
So to your first question, no, we haven't been able to measure specific muscle force yet. I'm really interested to see that because I would think that we're also preserving that, especially as the older they are, but we haven't been able to do that yet. So that's something that is on my pipeline. And then your second question, so we did the cognitive studies that I showed. We did the Barnes maze and the novel object on our aging studies. I don't have enough animals on my Alzheimer's studies to look at behavior yet, but that's also on the pipeline. And I also want to expand it beyond just those two. So we started with those two because they're more or less hippocampal dependent, and that's where we were seeing a lot of our benefits. But I would really like to look at anxiety measures on these guys. You know, the fear conditioning, you know, the water maze. There's a bunch of things. Like I have a list, and I'm just like, please get to the right age and give me the right number so I could just throw them at that battery of tests. Yeah. Very good question.
Thank you. Thank you for your question. And congratulations on your work during the COVID. It's amazing. Yeah, very challenging.
Thank you. So it looks like Dusty posted a question in the chat. Do you want to ask it, or do you want me to read it?
I can go ahead and ask.
Yeah.
I was just curious because I know that there are genetic markers for Alzheimer's disease, and some of the isoforms, it seems like, can prevent increases in BDNF that are usually induced by exercise. And I didn't know if you just had any plans to kind of parse out those specific isoform differences. I mean, because I know you said that you hypothesized that these are BDNF dependent. So I was curious to see if you were going to like explore.
Yeah,
deeper.
that would be a fascinating thesis project for an enterprising grad student or postdoc. But yeah, no, I currently don't have any plans for that, but I know that there's, God, the last time I looked, I think it was like 17 different isoforms of BDNF. And some of them are turned on under different conditions and in response to different stimuli, like you mentioned. And depending on what isotype you have, I think there's a particular SNP, Val 166 is for some reason is what's pinging in my brain, that makes you more or less, you know, responsive to some of these things. So our mice are all inbred, so they should all have the same one at least, but in planning to translate some of this to human populations, that's absolutely a question we should answer. Really good point.
Can I go back to your adipose tissue data for a minute too? I wanted to ask that question because isn't it like, it was so interesting to me that you had an increase in adipose tissue. Really? I'm sure you anticipated the opposite.
Yeah, I did.
So can you talk a little bit about that? So that is really interesting. And I was wondering if you did any mitochondrial assessments in your adipose tissue. I assume probably not, but.
Yeah, so we haven't done mitochondria. Essentially, I need, I don't know, like $10 million in a decade to actually characterize these mice fully. But we do save a lot of biospecimens because we keep finding those like interesting things, and I never know when a pilot or a collaboration is going to come my way. but you're absolutely right. I expected the opposite. And when, my student came to me, I was like, this is what I see. And I'm like, are you sure you didn't swap these? And so I don't know what is going on there. Some sort of lipid mobilization pathway is currently my, off the cuff, like this is complete speculation, is what's happening. I do know these guys don't eat more. We've measured their food
That was my
intake.
other question. My other question was, are they eating more?
Yeah, so they're not.
Okay.
And so one thing that I would like to see is these guys, because I'm running control and TFib animals too, is the running going to decrease that fat accumulation or not? Right? Because that's something you would expect in a runner.
What?
What?
Yeah, so I don't know yet.
You and I?
Not sure.
Go for a walk? We gotta
There was one other. Oh, the other thing I was going to mention is I wonder if your lower liver weight is due to reduced triglycerides, right? So
Exactly.
And
Yeah, so
kind of
like you're
along
moving
those
the lipids from one.
exactly, sequestering it in the fat, which is a healthy place. I study fat cells, so this is why I'm very
I see.
So this is my interest, and interest in the brain as well. So we have lots of commonalities. But I forgot where I was going with that. But yeah, so the insulin resistance piece. Did you look at systemic insulin sensitivity?
Yeah,
And
so
I would
those
imagine
same-
they'd be still good, right?
Yes. So and that was another thing where I was like, huh? And this is why we actually test things instead of assume. So
Yeah.
those same animals that I showed you, there's beautiful differences in the running performance and the mitochondrial function in the Ouroboros tests. We ran GTTs and ITTs on those. There's no difference whatsoever.
Interesting. But it's interesting that there's no difference despite the fact that I have more adipose tissue.
Yeah,
Because
exactly.
conventional wisdom would say, oh, well, maybe they're becoming insulin resistant, but adipose tissue is insulin sensitive too.
Yeah,
So
at least at the 2 ages we tested, we didn't see a difference. But yeah, absolutely. And so it's one of those things where you're like, yeah, of course they're going to have a different GTT. Nope, it's the same.
Interesting. And final question is, how do we increase our TFAB?
Exercise.
Okay, so exercise does do it.
Yes,
Okay.
it does. Yes.
Good deal.
Yes.
Good deal. Okay. All right. I'll stop monopolizing now. I'm sure other people have questions.
Yeah, I tell my students that
we're actually-
whoever
yeah.
graduates first is going to get a treadmill. That's going to be their gift. So go work out in the world.
Yes. Yes. Well, here in NEP, all of the faculty have treadmill desks. So we can,
I need
Yeah.
one of those.
yeah. So I was doing that actually while I had myself muted, but we don't use it all the time. I don't use it all the time, but it is nice to have.
I have a question.
Yes.
You talked early on about a muscle signaling brain, and could you expand on that? Is it simply molecules in the blood? Is it neural? I'm really thinking big picture here because I'm not in this field and I'm trying to get a sense of where the field is going.
Yeah. So as with most things in biology, I think it's a little bit of column A and a little bit of column B. So yeah, as you pointed out, the muscle is directly connected to the central nervous system via the neuromuscular junction, right? And there is evidence of things that can retrotransslocate from the muscle back into the central nervous system. So I have no doubt that there's something happening there too. But there's also this idea of the circulating factors that I think, seeing the benefits of exercise materialize in a sedentary animal with just a plasma transfusion is like astounding to me, right?
Yeah.
And so are they the exact same benefits? Probably not, right? And do you need, again, a little bit of column A and a little bit of column B to get the full benefits? Probably. Absolutely.
Yeah, and my sense is the brain is pretty restricted as to what it lets in. And so if you have candidates, you need receptors, you're going to need to be able to get them in there. And actually people have tried, and it's very difficult to see even fatty acids cross the blood-brain barrier. So anything that's fat soluble, I need to revise the way I think, because if many things are coming in from different organs and signaling the brain, then it's not the closed box that I think it is.
Yeah. And that's the black box in the field right now, right? So it's been demonstrated that some of the factors that I showed in my introductory slide, if you just deliver recombinant cathepsin B in the periphery, you see the effect in the brain. If you deliver, I think it's AAV based, but same idea in the periphery, the GLPD1, which is the liver based factor, you see the effect in the brain. And so necessarily, from the therapeutic standpoint, we don't even need to figure out the mechanism yet, as long as it works, right? And like you said, the brain is so protected behind the blood-brain barrier, but there is something about this signal that gets through. And so we don't know if it's signaling at the blood-brain barrier, and in particular, somebody mentioned neuroinflammation. So the astrocytes have their feet sticking in the blood-brain barrier, and they could actually be direct targets of this. And then they transmit the signal in. So something doesn't necessarily have to cross it. It can just hit right there and essentially propagates inside.
Yeah, thank you. Thank you for those ideas. And for such
Dr. Cortes, we are at time. We're 5 minutes after 5. So I want to be mindful of that. I know some people have to run and get kiddos, things like that.
Yes.
Would it be okay? I think some people may still have questions. Can we email you?
100%, please. Yeah, you have my Twitter. I think I had it on my slides. You can email me. I love to talk about this, especially for trainees. If you guys want to talk collaborations, I'm junior faculty, so I receive all the things.
Perfect.
But yes, thank you so much for the invitation. This was lovely.
Thank you so much. We really enjoyed it. So
Yeah.
It's such a great talk.
Thank you, Japna.
take care, guys.